The Gabaergic deficit hypothesis of major depressive disorder. Major depressive disorder, also known as MDD, is a very common mood disorder that is associated with constant feelings of sadness and the loss of interest in daily activities. There are different factors that can affect the likelihood of MDD in people, including major life changes, genetics, side effects of illness or medication, and social or environmental stress. Because MDD is a complex disease, many hypotheses about what actually causes this disease have been investigated. Recent advancements in neuroscience suggest that there is a relationship between MDD and lowered concentrations of GABA in the brain. This is known as the Gabaergic deficit hypothesis of MDD, and it provides key connections that aid in understanding MDD's effect on the brain. So what is GABA? GABA is a key inhibitory neurotransmitter or signaling molecule in the brain. It plays a large role in sleep regulation, memory, mood, anxiety, and stress. When GABA is reduced, it can lead to these processes becoming abnormal and lead to issues such as insomnia, chronic stress, and MDD. GABA has multiple receptors, but the main type of receptor involved in this hypothesis are GABA-A receptors. GABA-A receptors are GABA-gated chloride channels composed of 5 different subunits that form a pore, and they are located on postsynaptic neurons. When GABA binds to its receptor, the receptor channel opens and chloride ions are able to flow into the cell. In order for a neuron to pass a signal to neighboring neurons, its membrane's overall charge, or membrane potential, must become less negative. Since chloride is negatively charged, its entry will cause the cell to become more negatively charged, and therefore unable to pass further signals onto the next cell. This is technically known as hyperpolarization. The Gabaergic deficit hypothesis of MDD states that reduced transmission of GABA may be a key cause of mood disorders such as MDD, and that treatment with monoaminergic antidepressants can reverse these effects. The cortex of the brain is normally inhibited by GABA interneurons. Once this inhibition is lost or reduced, MDD can occur. Reduced GABA transmission leads to the frontal cortex becoming overexcited, exciting the neurons of the hypothalamus as well. These hypothalamic neurons are part of the hypothalamic pituitary adrenal axis and can induce a stress response when excited. So, reduced GABA transmission ultimately results in an increased stress response. Evidence for this hypothesis includes research that has found reduced GABA concentrations in MDD patients' brains, as well as alterations in GABA-A receptor subunits that make the channel less efficient to let chloride ions flow through. Another important player in this hypothesis is brain-derived neurotropic factor, or BDNF. BDNF is a biomolecule that plays important roles in the formation, function, and survival of neurons. If BDNF signaling is disrupted, this can lead to neurological disorders, including MDD. In fact, research shows that BDNF concentration is reduced in MDD patients along with GABA. BDNF is important in the Gabaergic deficit hypothesis because it regulates GABA transmission. Increased production of BDNF will lead to increased release of GABA. In immature granule cells, when GABA binds to its receptor and allows chloride ions to flow out of the cell, this causes the membrane potential to become more positively charged. This change in membrane potential will facilitate calcium entry via voltage-gated ion channels. Increases in calcium will result in increased activity of protein kinases, such as pKa and CAMK2, that phosphorylate and activate CREB, a transcription factor that increases the production and release of BDNF. BDNF can then go out and act on GABAergic terminals and promote the release of GABA. The study shown here was done using immature granule cells where the direction of Cl flow is outward. However, in mature granule cells, the direction of Cl flows is inward. In immature neurons, enhanced BDNF production was shown to enhance GABA release in the GABAergic terminal. If this holds true for mature neurons, then the downstream activation of CREB would increase the production and release of BDNF, therefore promoting the release of GABA. In MDD patients, there is a GABA deficiency, so there may not be enough GABA to bind to GABA-AR and trigger the entry of calcium into the cell to start this entire process. But luckily, SSRIs can work to rebalance this process in MDD patients. MDD is often treated with selective serotonin reuptake inhibitors, or SSRIs. Serotonin is released into the synapse, and it normally re-enters the cells shortly after due to the serotonin transporter SERT. However, SSRIs work by binding to and blocking the SERT to increase the concentration of serotonin in the synaptic cleft. There is evidence suggesting that when MDD is treated with SSRIs, there is a downstream effect on Gaba. SSRIs work to prevent serotonin from leaving the synapse. When there is a higher concentration of serotonin in the synapse, the likelihood of stimulating 5-HTRs, which are serotonin receptors, increases drastically. The stimulation of serotonin receptors in mature cells will result in the activation of cyclic AMP, a second messenger. Cyclic AMP then goes on to activate CAMP protein kinases, or pKa. pKa will then go on to phosphorylate CREB. CREB then escalates the production and release of BDNF, which will increase the release of GABA. The addition of SSRIs to this cycle allows for it to continue working functionally. Although not all of the mechanisms involved in this hypothesis are precisely known, the Gabaergic deficit hypothesis of MDD still provides researchers with key connections that can lead to new discoveries and treatment options for MDD. Along with major depressive disorder, GABA deficits may also play a role in schizophrenia. However, it is still unclear if GABA subunits are altered in vivo or are related to the symptoms of schizophrenia. So how can reductions in GABA cause both major depressive disorder and schizophrenia? How are MDD and schizophrenia related? The answers to these questions could be obtained through studies using proton magnetic resonance spectroscopy to study GABA levels in certain parts of the brain and could provide insight on how GABA is affected in both of these diseases. Measurement of GABA levels in vivo by means of MRS offers the possibility to approach both illnesses from a unique perspective and provides insights in the relationship between GABA deficits in both MDD and schizophrenia. However, future MRS studies using GABA editing are required to give us a better understanding of schizophrenia and MDD in different stages of the disease. GABA editing at ultra-high field strengths will be beneficial for detection of the relatively small GABA signal, because of the increased sensitivity and resolution, which allows for an accurate and time-efficient assessment of GABA levels. We hope you enjoyed this presentation, and thank you for listening. The Gabaergic deficit hypothesis of major depressive disorder.